welcome to everyone to this second event on this Instruct webinar series. And these seminars are presented by the Instruct Italy Center. And so then presenting the results of the activities performed in Florence at the Instruct IT Center. And here there is, this is a picture of our center, where you can see that we are a major NMR facility with quite a number of NMR instruments. We have 12 NMR spectrometer from solution, solid state NMR. Most of them are equipped with cryoprobes. And in particular, we have the first available 1.2 GHz NMR instrument, and they actually probably the only one in operation now and open to users. And we have really the largest available magnetic field range, because we have also a relaxometer. We can collect the measurements at very low frequency. And all these instruments are also integrated with pulsed EPR, that is another magnetic resonance technique, very powerful for characterizing interactions and paramagnetic centers in biomolecules. And in general, we have a new cryo-EM instrument just installed, mass spectrometry, obviously molecular biology, cellular biology labs. And here is the picture of this 1.2 gigahertz, these large instruments, and you'll see some results already collected and spectra collected on these instruments. And as you can see from understand and grasp from the program of today, there is a variety of activities which are performed in Florence as research, which goes span of the various possible areas, going from in vivo to in vitro to in silico. And so then we characterize NMR directly in living cells. So we do EPR in living cells. We do drug screening in living human cells. Obviously, there is the core activity of structure determination with molecular biology labs, obviously cellular biology labs. We do drug screening and drug design, vaccine design as well, functional characterization of processes, protein-protein interactions. And there is a very large group of researchers on metabolomics of biological fluids as well as on food. Another important pillar of the activity in Florence is the in silico activity, which spans from bioinformatics, computational biology, and advancement in methods, new experiments, and theory for NMR. And so then magnetic resonance in general. So we are really spanning different areas and different competencies. So this is the program of today. Now Dave will give a presentation of Instruct-ERIC.
So I hope that people in the webinar will know about Instruct by now. Instruct, we've been running a few years. We are the sort of pan-European infrastructure to provide access to cutting-edge methods in structural biology, and do training and encourage people to move around and do internships when people can move around. And we hope that that will be coming back. But the key thing is that the provision from Instruct is from the centers, and we're getting more countries joining Instruct. We're getting, we recently had our first international infrastructure join, which is EMBL, and their centers will be coming online. So the idea of these webinars is that they move around from center to center. The first was in the Weizmann in Israel, the second in Florence. There'll be another one coming up before long in Holland, and the idea is that this will give an opportunity to showcase the science that's done and also some of the sort of new technologies. I think it's important to do this because the democratization of science, opening up these cutting edge techniques to everybody is the whole sort of raison d'etre for Instruct, to enable us to really maximize the impact of structural biology scientifically. For people that have not used any infrastructure in Instruct, I'd recommend you to look. It's very easy to apply. A lot of the facilities are now available remotely, so it can be accessed without traveling. And this is a lot of activity ongoing. And I hope these monthly webinars will be a way of people keeping in touch and getting a bit of encouragement in what can be very difficult times. I know. Thank you.
Thank you, Dave. Thanks a lot. First speaker of this webinar is Enrico Luchinat, and he will talk on in-cell NMR drug screening through in-cell NMR. Please, Enrico.
Thank you, Lucia, for the introduction. Thank you to the organizer for giving me the chance to talk to this webinar and show some of our results. I'll talk about some of our most recent results of drug screening by in-cell NMR that we obtained here in Florence. I'm working here since a few years, and together with Lucia, we've been doing in-cell NMR since a few years by now. So in-cell NMR is a branch of biomolecular NMR spectroscopy, which exploits the high resolution of the modern high-field instruments to obtain directly atomic level information on macromolecules or small metabolites directly in living cells. So it's a very direct way to obtain such a high-resolution information in a physiological environment. So it's a powerful tool to get structural and functional information in cellular context. And that can be applied, and we applied it years ago to study protein folding and maturation events in human cells. And it's also been applied elsewhere in the world to probe confirmation of unfolded proteins involved in diseases such as alpha-synuclein, and it can even be used to resolve structures ex novo. Now, the way we do it here in Florence, we adapted a protocol for high-level protein expression in human cells, which was originally developed in Oxford, and it's part of the platform for mammalian expression. We also have that kind of platform for a small scale protein expression in human cells, which can be accessed by Instruct users, by the way, with the purpose of doing in-cell NMR here. And that's based on a highly efficient transfection protocol. We use HEC293T cells, which are human-derived, and they're very robust cells that can be transfected to high levels with many copies of DNA and can reach high levels of protein expression. So we exploit the cells themselves to produce the protein of interest, to increase the levels which are necessary for NMR detection, and we can also then label these expressed proteins with the isotopes that allow multidimensional high-resolution NMR if needed. And that can be used to obtain even more structural information. Then cells are collected and analyzed in the high-field NMR spectrometers. So this has several advantages because, for example, we do not require too much manipulation of the protein sample because the cells are basically expressing protein. And so that even gives access to sensitive proteins which may be degraded or prone to aggregation or sensitive to oxidative modification, for example. Then more recently, we sought to apply this methodology to study, to basically improve the efficiency, that's the hope, of the drug development pipelines, which are very expensive, slow, and sadly inefficient sometimes, because we go from the in vitro screening of possibly in silico screening of millions of small molecules, and then lead optimization of thousands of molecules, and then in vitro binding assays then cell-based assays, and then ADMET, so activity and toxicity studies. And there is a very high attrition rate where you basically lose very good candidates in the later stages because they may not be able to reach the target. If there's an intracellular target, they may not be able to reach it, or they may give rise to toxic unwanted side effects, which may be due to the not high enough specificity of binding to the wanted target. So we sought to find out whether, I mean, we believe that the SSNMR has a great potential in this and could fit between the screening in vitro, which is high throughput screenings very often today, and the preclinical studies on cell models or animals to improve the success rates of the candidate drugs. So we focused on a protein which is very well known since a long time, which is human carbonic anhydrase II. Which is now we focused on the second isoform, but there are 16 isoforms in humans, which are fundamental for pH regulation and homeostasis, and CO2 regulation, and also sodium transport in several tissues. So they are involved also, the dysregulation is involved in a lot of pathologies, like glaucoma, epilepsy, and cancer. So this is very well known, but it's a very important target yet nowadays. It's a metalloprotein with a zinc and its active site, which is coordinated by 3 histidines. And it is amenable to study via solution in cell NMR in human cells. This is a 2-dimensional spectrum of carbonic anhydrase II overexpressed in human cells in 2 forms, in the free form, so without any ligand, and then with a ligand bound to it. And then we can appreciate that the spectrum is changing in the cell, which means that the structure is perturbed, which means that we can monitor ligand binding directly in the cell. Now, even more interesting for our purpose is the fact that the signals from these protons of the histidine ligands, the zinc ligands, they fall in a region of the 1D proton NMR spectrum, which is basically free from any other signal arising from the cells themselves. And that means that we can skip the nitrogen-15 labeling and just produce cells with Ca2 expressed in it and observe monodimensional spectra where which contain a signature of the molecules bound to the protein. And different molecules gives rise to different chemical shift perturbations. So it's even possible to distinguish the free protein from the bound protein. Then we went on and exploited this this NMR approach to monitor the binding of a series of compounds, including some reference compounds, which are 2 of which are actually approved drugs against glaucoma, and then a series of experimental ligands which were developed by Claudio Super and group here in Florence. Then we proceeded to monitor the binding, treating the cells with these compounds, with a high excess of compounds, and then observing the spectrum of the protein inside the cells. And we immediately realized that a few of these ligands were binding very efficiently, 100% binding protein, whereas several others just gave no effect on the spectrum, which meant that we still observed the free protein. That did not correlate immediately with the Ki, with the inhibition constant in vitro of these compounds. So that meant that there was something else in the way between the protein and the ligand. There was something preventing the binding. Then we focused on those compounds that bound at high, in excess doses, could bind 100% the protein, and we started doing a dose response analysis. Then we also did a time response analysis to assess whether at which concentrations we still got binding or partial binding. And as you can appreciate, we actually went from no binding to mixed states and to 100% binding. And these spectra could be analyzed by deconvolution to separate the 2 species, the free and the bound protein, and that allows us to quantify the amount of bound protein versus free protein, and therefore obtain dose and time response binding curves. Those were then fitted. We made a few hypotheses on which kind of mechanism could be at play. And the most simplistic model is a diffusion limited kinetics, where the plasma membrane is actually slowing down the diffusion of the molecule to the cytosol, followed by fast binding, which was hypothesized due to the very low KD, so the very high affinity. So then we modeled that with equations, which included also a time dependence, which was due to the diffusion through the plasma membrane. And those would basically fit quite well the data, both in the time dependent and the dose dependent curves. So with that, we retrieved a measure of an empirical measure of the permeability constant through the plasma membrane, which was, and that was an interesting result, because we found a striking correlation between the in silico prediction of the permeability expressed as log Kp, and the actual binding or lack thereof in our experimental conditions. So we observed that several compounds which were not binding, they had actually a much lower permeability, predicted permeability, which was corroborating our hypothesis. And then even more strikingly, the two approved drugs had different permeability in our conditions. They differed about 1 order of magnitude, which correlates with the dosage recommended for these two drugs in the treatment of glaucoma. So that is a very good indication that we actually are probing some physiological behavior that is relevant then in the later steps of a drug discovery pipeline, where you are interested in maximizing the permeability, because that would also increase the potency of the drugs. So then we moved on and started investigating with the same approach another series of compounds, which this time are approved drugs, some of which, all of which were selected because they are known to inhibit carbonic anhydrase. So these are a few compounds. These are the compounds that we selected for this study. The property of a few of them is that they show off-target binding activity. They are known to inhibit CA, but they were not developed on purpose to inhibit CA. So first ones are COX-2 selective inhibitors, so they are anti-inflammatory drugs. Then we selected a few inhibitors of ion transporters that are used as diuretics. Then an anti-epileptic drug, which also inhibits voltage-sensitive ion channels. And then 3 more compounds, which were actually developed to, where the main mechanism is the inhibition of carbonic anhydrase. So they are employed to treat glaucoma, and one of them is actually under trial as an anti-cancer drug because it inhibits the isoform of CA, which is involved in cancer. And then we employed the same, so these all inhibit CAs, they are known to do that in vitro, but none of them have the CA as a primary target. And then we found this, that when treating the cells with high dose of these compounds, some of them showed complete binding, like the previous study, and some others showed incomplete binding. These data were collected over an average, time average in 30 minutes after cell treatment for 1 hour, then cell collection, and then treatment, and then measured by NMR for 30 minutes. The binding was not complete for many of them. So in the spectra, you can see the free form still present, and this is the result for these compounds. Now, interestingly, these the compounds which bound fully bound CA were those 3 that were known to actually inhibit CAs as their primary target. So there had to be a correlation with that. We further analyzed the dose dependence, the time dependence of these compounds, and then we also performed time-resolved NMR analysis directly in the tube. So by performing several short experiments in a 30 minutes, in 1 hour time, we could, with these data, we could divide these compounds in 3 different classes. The first one was the most obvious, the fast diffusing, strong binding compounds. which even at much lower doses would still show 100% binding. Then a second class where the binding fraction was clearly dependent on the treatment, on the time of treatment in the incubator. So we just needed to wait longer to let this compound permeate the plasma membrane of the cells. And then a third class of compounds, which at time zero, right when we started measuring by NMR, they were actually found to be almost 100% bound. So these are plots of free Ca2 versus bound Ca2. And you can see that in about 1 hour of experimental measurement, the bound fraction decreases and the free fraction increases. Now we also checked that this happens in the incubator, so with attached cells in the CO2 incubator. That means it's not an artifact of our experiment. It's really something that when you remove these ligands, they somehow unbind from the protein. And that is interesting because we still have to figure out what could be the mechanism. It's not something that it's obvious or straightforward to tell, but there's an interesting correlation with the fact that these drugs are known to bind to different targets in cells. And then finally, I'll just finish here, but I'll show you some other development that is ongoing here, which is also part of Instruct Ultra and is founded by an Instruct JRA award, which is the development and the implementation of an improved bioreactor designed for SSNMR experiments. Now, I think this is really promising and is part of our effort to also make this available to users of Instruct in the future. That allows us to to keep cells alive, viable, and metabolically stable in the NMR instrument for hours up to days, up to 72 hours. We've measured live cells with ATP synthesis going on up to 72 hours from detachment. And that is extremely important because it allows us to record longer NMR experiment, but most of all, it allows time-resolved NMR. So with this setup, which was developed with the great support of Matteo Pennestri from Bruker, it's basically composed of a flow unit which replaces the growth medium of the cells. The cells are entrapped in the spectrometers, and there's many ways to entrap them. We use the hydrogels to keep the cells inside the NMR. And that is what you get when you treat the cells with the ligand. This is, again, cells expressing carbonic anhydrase II. So these are the histidines of the active site of the protein. And then you get a time-resolved binding curve. Now, these can be quite easily analyzed in a quantitative way by using a multivariate curve resolution algorithm. So it's something which is, for example, already implemented in MATLAB. And so we use this workflow, which is semi-automated, to obtain binding curves which quite a high time resolution, which are able to separate the free from the bound protein, and that they can therefore be fitted with kinetic equations to retrieve the kinetic constants, and then therefore understand the behavior of ligands and macromolecules in principle in a cellular setting in a time-resolved way. So with this, I would like to thank all the people involved in these studies, and thank you for the attention. And now I hope there are some questions. Thank you again.
Thank you, Enrico, for this presentation. Yes, there are many questions. There is a question is asking how easy is it to do isotope labeling in mammalian cell, and also is preferential to have an active site with histidines just to follow the binding the cell, or other amino acid are as good. And finally, do you need to have spectra of isolated protein or its structure?
I'll start from the last one. That's easy. So yes, it helps a lot to have the spectra of the isolated proteins. So it always helps. The more data you have in vitro, the better, because with in vitro you have more possibilities. You get more data. You do more NMR type of NMR experiment. It's really helpful to have in vitro data to compare against. Then concerning the labeling is quite straightforward. because you just replace the medium during protein expression. However, it could be expensive depending on the type of medium that you use, because these proteins, these cells need all amino acids labeled. You cannot you cannot do that with bacteria. So it can be expensive. You can choose, you can do residue selective labeling, so that cuts down the costs and also cleans the spectrum. So that's linked to the last question, do you need histidines? Then this is a lucky case in a sense, because we can skip the labeling. However, in principle, you just need to label whichever signal is perturbed by the binding to do this kind of experiments. So you can also label different amino acids. You don't need histidine.
Okay. Other question, if you don't isotope enrich the protein of interest, how much excess of protein do you need to get the signal over the background? And also he commented, great talk.
No, thanks. Thanks a lot. Well, this is it's a very good question, because unfortunately NMR is quite insensitive. So right now we still need to overexpress the protein irrespective of the labeling. So we could not observe the endogenous carbonic anhydrase, even if its signals would fall in a clean region of the spectrum. So we still need to increase the levels. Then, if the signals are well resolved, then you would not need the labeling. So in this case, for example, that I showed. In any other case, then you wouldn't need the labeling, and you will still get some background due to the labeling of other cellular components, but still the overexpressed protein will pop out of the spectrum. And then we can also further process the spectrum by using a control spectra on cells which are labeled but do not express the protein of interest, to subtract the background, and that even improves the selectivity of observation. So we can really see, labeling really helps to observe other signals which fall into crowded regions of the spectrum.
Okay, good. Thanks. Other question, wondering how do you quantify the protein and then consequently the ligand concentration that you add?
Yeah, that's another good point. We need to do quantitative assessment of both things, of course. So we quantify the protein by running gels on the cells. That is, we make a cell extract from the whole flask of cells, which is the same amount of cells that we put in the NMR. And then by quantifying the protein by gel, or we could also do that by Western blot if the gel was not good enough for quantification, then we can calculate how much molecules of protein are there in the flask, which is the same closed volume in which we add the ligand, and then we know how much ligand we add. So we know the molar ratio of the two. And we know the total volume in which the ligand is added. What we estimate is then the total cell volume, which is just an estimate because it's hard to tell apart the volume occupied by the cytosol versus the volume occupied by everything else. However, that is, it turns out that it's not really necessary for a proper calculation with the formula, with the math that I showed before. So you just need to know the molar ratio.
Okay, there are several questions more. Is it possible to triple label in human cell lines?
So double label can be done, of course. Triple label would require growing the cells in D2O. That can probably be done to some percentage of D2O. However, then so in principle, yes, you could deuterate by using a deuterated growth media with deuterated amino acids. It's not something we've ever done, but it's probably a matter of how well the cells would survive in highly deuterated media. So for deuteration, this could probably be trouble. It's probably much better if you deuterate just one or a few amino acids and then express the protein still in water. So that would partially deuterate different protons in side chains.
Two quick questions. Could the ligand-based technique be applied?
That's something we are also investigating right now. You need some way to observe only the ligand, and with proton detection, that's currently impossible. But there are other nuclei, for example, fluorine-19, that can be used for that purpose.
And are you fixing the cells while acquiring the data, or they are metabolic active?
Not at all. They are metabolic active. So we just take the cells from the flask and then make a pellet and then or put them in the bioreactor. In the bioreactor, they're fully alive and metabolically active, so we don't fix them.
Okay. Thanks again, Enrico. And
thanks
then
to you and to everybody.
Yeah, and then we go on with the second presentation by Francesca Cantini, and she will talk on the usage application of NMR and the 1.2 GHz applied for the characterization of SARS-CoV-2 proteins.
So good morning, and thanks for inviting me to the second Instruct-ERIC webinar series. I would like to show some new results obtained on SARS-CoV-2 protein with 1.2 GHz. As already Lucia mentioned, the CHERM is now equipped with the new 1.2 GHz instruments, and the arrival of the magnet was practically coincided with the arrival of the virus, SARS-CoV-2 virus here in Italy on February 2020. Indeed, it was installed during the lockdown here in Italy. And in that period at CHERM, we immediately wondered if this new generation of magnet could have a significant impact on the development of NMR spectroscopy and its application on structural biology in general, and in particular on the characterization of SARS-CoV-2 biomolecules. As you know, in March 2020, the World Health Organization declared the pandemic of COVID-19 due to SARS-CoV-2 virus. And all of us know that, unfortunately, COVID-19 lacks effective therapeutics. Several drug targets are in the spotlight for the treatment of SARS-CoV-2 infection, and also no drugs, no vaccines are available related also to the previous coronaviruses such as SARS-CoV-2 and MERS. Within this frame, we ask ourselves, how can NMR spectroscopy support the research against COVID-19? And we think that the new 1.2 GHz instrument may be an excellent occasion to zoom in into atomic details of proteins and RNA involved in SARS-CoV-2, with the goal to identify a strategy to interfere with it. Contemporaneously, at the end of March 2020, an international research consortium was founded at Goethe University of Frankfurt, and in Frankfurt, and CHERM is part of it. And the goal of this consortium is to support the search for antiviral drugs by enabling a NMR-based approach. This required the large-scale production of druggable proteins and the RNA of SARS-CoV-2, and also the extensive NMR assignment of the NMR assignment of the proteins and RNA and their structural determination. And this is a prerequisite for rational drug design. And the SARS-CoV-2 proteome is about 30,000 nucleotides long. It encodes for 28 proteins, 4 structural proteins, spike protein, envelope membrane, and nucleocapsid protein, essential for the viral assembly, about 8 accessory proteins, and 16 non-structural proteins with different roles. The moment in CHERM, we focused our attention on non-structural protein 3. This is a multi-domain protein, and each domain is divided by flexible linkers. Each domain has different roles. And we've focused our attention in particular on NSP3B. This also known as ADP ribosome binding protein or microdomain. Is a conserved ADP ribose binding microdomain. The microdomains in SARS and MERS are thought to play a key role in viral replication, and they are structurally conserved and are thought to enzymatically remove the ADP ribose from proteins. The ADP ribose is a post-translational modification, and so this function, so the DADP ribosylating function of microdomains of NSP3B has evolved to protect the virus against the antiviral ribosylation catalyzed by polymerases. So for this reason, also the microdomain NSP3 became a highly relevant drug target because it's involved in this process. Here I am showing you the HSQC, 59 HSQC experiment acquired on 1.2 GHz. The macrodomain NSP3B is 173 amino acid long, and all the expected spin system are present in the HSQC experiment spectrum. And here just briefly I would like to show a zoom in a 1D experiment of the same acquired on the same protein sample at with the same parameters at the 700 MHz and 1.2 GHz. As expected, the line width is extremely decreased in the spectrum acquired at 1.2 GHz, but also, the resolution is much higher in the experiment acquired at 1.2 GHz. All the NMR experiment for backbone assignment of SARS-CoV-2 NSP3B were acquired at 1.2 GHz. Here I have shown here F1 free plane of HNCO where all the expected carbonyls are present. And this is true also for a less sensitive experiment, which is the HNCA-CO, for which almost all the expected carbonyl are visible. The APO form of NSP3B was the assignment of the APO form was almost all complete. We assigned the 99% of NH backbone pairs, and all the COCACB chemical shift were assigned. The assignment was deposited on BMRB, and the assignment, thanks also to the high resolution and high quality experiment acquired at 1.2 GHz, was done in few hours. On the contrary, in the case of the ADP ribose bound form, we miss about 20 residues in amide resonances, here shown in pink. And because in the HSQC experiment, we the amide resonances are brought beyond detection upon addition of the natural ligands. Anyway, we were able to fix, to map the binding site, the ADP ribose binding site within the crystal, the structure, and the binding site was in very good agreement with the recently solved crystal structure of the ADP ribosome bound form. These data are very important because they represent the starting point to further studies, and the obtained NMR data will provide the basis for the NMR investigation at targeted at obtaining the small biomolecule inhibitors of NSP3B, and moreover the crystal structure of the ADP ribosome bound form help us also to set up some peculiar experiment of 1.2 GHz, the carbon nitrogen filtered NOSI. This experiment can be exploited to obtain structural restraints between the unlabeled ligands and the labeled protein, in order to find the position, in order to fix the ligand within the binding site. Here, this is the F1, F3 plane of the carbon-13 filtered NOSI. Here, this is the ADP ribosome signal, while this is the resonance, the frequency of the methyl group of a valine, that is located in the binding site. And in the next future, we will use the assignment information and this kind of experiment to screen also different compounds in order to find a small molecule able to bind and interfere with the NSP-B activity. So in order to apply this kind of experiment, we need the complete sidechain assignment of all proton resonances. And just to conclude, I would like to show a very high resolution experiment of here in this is a so fast experiment of a methyl where we observe the methyl resonances, and only the methyl resonances in a double level sample. And the resolution achieved at this spectrometer, we think that can be beneficial in the structural characterization also for in the viral and viral host protein-protein and protein-RNA interaction. And also in comparable studies, if you think that this kind of experiment can be acquired also on unlabeled, for example, antibodies, and also, for example, on in the future or antibody elicited elicited by the SARS-CoV-2 virus. And with this slide, I conclude, and thanks all for your attention.
Thank you, Francesca, for this presentation. I start with, there was a question in the chat asked, did you use deuterium labeling?
No. This is, the protein is 170 amino acid long. We don't need the deuterium. So we acquire, all the experiments were acquired on a double level sample.
And there is questions, the decrease in line with this protein at ultra high field versus lower field could be due to exchange broadening?
No, I don't think so. The experiments were acquired exactly on the same sample. So you mean exchange broadening between a free and bound state, but I think...
Or different conformations.
But between 700 and 1.2 gigabytes, yeah. The sample is exactly the same. We acquired it with the same, so I mean, it could be, but we see this effect now I have shown only one region, but this effect is here for all the experiment, all the from 12 ppm to minus 3 ppm, also in the aliphatic region. So I think it's more due to the fact that we are acquiring the experiment at 1.2 gigahertz than from exchange broadening. So it's an effect of the Eiger field.
If no other questions, let's go on to the third presentation that is presented by Marco Fragai, and we will talk on the application of solid-state NMR for the characterization of functionalized biomolecules.
Of the protein component in heterogeneous material, like, for example, bio-inspired materials, or when the protein are conjugated with polymers, or when they are grafted on nanoparticle or absorbed on adjuvants in a vaccine formulation, is still a challenge for structural biology. Here at CHERM, we have developed strategies for the use of solid-state NMR for the characterization of these difficult systems. Concerning bio-inspired materials, a protein can be easily embedded in a silica matrix by adding to the protein solution the tetrahydroxyorthosilicate and then an external templating reagent, for example, polylysine, or the biosilification process can be catalyzed directly by the protein if you use a protein with this suitable fusion peptide. And we have here, we have collected, using obviously an isotopically enriched protein, we have collected, this is an example of the catalytic domain of MMP12 embedded in the biosilica. So we have collected the solid state experiment on these biomaterials. And here we have a comparison of the bidimensional carbon-carbon DAR spectra collected on a protein embedded in polylysine compared with the same spectra collected on a crystalline sample of the protein. And we can see that the quality of the solid state spectra and the resolution of the spectra collected on the bio-inspired material is similar to that collected on the crystalline sample of the protein. Quality of the resolution is so good that the solid state spectra can be used to reassign the resonances and to, for example, assess the preservation of the protein folding within the matrix, and also to identify the region of the protein experiencing structural changes. The quality of the spectra is similar also if you use a protein bearing a self-templating peptide. And we have collected different kind of solid state spectra. For example, here you can see the hydrogen-nitrogen cross-polarized HSQC spectra collected on a silica matrix embedding a catalytic domain of MMP12 with the self-templating peptide, but also the quality is good also on the, for the spectra collected on the green fluorescent protein. And this kind of spectra can be directly compared with the HSQC of the protein collected in solution. The quality and the resolution is so good that, for example, we are able to monitor the interaction of the protein embedded in the matrix with the ligand. This is an example of the carbonic anhydrase. And so it is possible to identify the residues experiencing the chemical shift variation due to the interaction with the inhibitor. However, the solid state spectra can be used also to characterize, for example, different systems. For example, protein conjugated with polymers. In particular, we have investigated the PEGylated proteins. Why PEGylated proteins? Because many biotherapeutics are PEGylated proteins. And why PEG? PEG is useful because the conjugation with the polyethylene glycol chain improves the stability of the protein, improves the water solubility, increase the circulation time by reducing the renal clearance. This is important for small protein, for example, but more important, prevent the uptake by the reticulous endothelial systems. Moreover, the PEGylation prevent the hydrolysis of the protein by the proteolytic enzymes that are present in the bloodstream and the extracellular fluid, and also shield the protein from the immune system. However, PEGylation increases the size of the protein and the hydrodynamic volume, make it impossible, in many cases, the use of solution NMR for the characterization. Moreover, PEGylation hampers the crystallization. In our studies, we have decided to focus our research activity on a real case, the E. coli asparaginase. E. coli asparaginase is an enzyme that is approved for the clinical use against acute lymphoblastic leukemia as a native protein and also in its PEGylated form. It's a very big protein, is a tetramer of 140 kilodalton. So, first of all, we have proved that PEGylated protein provide solid state spectra of really good quality. Here we have a comparison of bidimensional carbon-carbon DAR spectra collected on the crystalline sample of superoxide dismutase, on ferritin, and on a crystalline sample of asparaginase. On the right, in blue, we have the same spectra collected on a PEGylated sample of superoxide dismutase, ferritin, and asparaginase. The quality is similar. And for SOD and asparaginase, the spectra collected on the PEGylated sample and on the crystalline sample are almost superimposable. This proves that the protein preserves their native folding also after the PEGylation. However, we decided to go in more detail in the characterization of the PEGylated protein. However, to do this, we have integrated the data collected by solid-state NMR with the data collected in solution. And we have also performed nuclear magnetic resonance dispersion profile on the native protein and on the PEGylated sample. And we have also used the X-ray data, and we have collected also cryo-EM images on the PEGylated sample. The first step was the assignment of the resonances and of the spin system of the PEGylated protein. So obviously the spectra collected on the PEGylated asparaginase are of good quality. So we have collected a set of spectra to assign the resonances. And so finally, we were able, integrating all the data collected on the native protein in solution and on the native protein used as a crystalline sample and on the PEGylated protein, finally, we were able to assign 236 spin system of the PEGylated protein, about the 74% of the total. So this is a really big protein, so was not easy. And we were able to assign 19 of the 22 lysine residues. The assignment of the lysine is important because the conjugation occurs on the side chain of lysines. So this is the cryo-EM images collected on the PEGylated asparaginase, were collected at the Nissen in Leiden, and the images prove that the protein preserves its tetrameric structure. And then after, with the assignment and collecting carbon-carbon DAR spectra at different mixing time, we obtain the intra-residues restraint and long-range restraint that were used in a docking calculation to generate a structural model of the PEGylated protein. Using similar strategies, we decided to see if solid state spectra could be obtained also on protein conjugated with the polysaccharides. Why is so important the glycosylated, so important the glycosylated protein? Because many vaccines, for example, the vaccines against Neisseria meningitidis, are obtained by conjugating the capsular polysaccharides of Neisseria to an immunogenic protein. However, the isotopically enriched mutant of the diphtheria toxin was not available. So we decided to, in collaboration with GSK, to use as a model the asparaginase that was conjugated with the capsular polysaccharide of MenC, of the strain MenC. And the glycosylated protein was used to, to collect a set of solid-state NMR experiment. And finally, we were able to reassign the lysines that where the conjugation occurred. And finally, we were able, by analyzing the spectra, to provide a semi-quantitative evaluation of the conjugation pattern of the protein. This is really very important because the immunological properties of glycosylated vaccines is related to the conjugation pattern. And so this kind of analysis is obviously difficult, but can be useful for the quality control. Then we have investigated a different system. So concerning, again, vaccines, we have proved that solid-state NMR experiment can be used for the characterization of a protein absorbed on the aluminum hydroxide adjuvant in a real vaccine formulation. Obviously, also in this case, a isotopically enriched protein is needed. So we use against our model, the asparaginase, again in collaboration with GSK. They use our protein for the preparation of the vaccines model, and then we perform experiment on this vaccine formulation, solid state experiment, and we obtain spectra of good quality, although the amount of protein is not very high. But finally, we were able to reassign most of the resonances. We were able to prove that the protein in the vaccine formulation preserve its native folding, and this is really important to understand the mechanism of the vaccine. And then we were able to identify the residues that experience the largest effect due to the interaction with the vaccine components. And the last example that I would like to describe is the capability of solid-state NMR to investigate protein grafted on the nanoparticle surface. So we have first of all use PEGylated gold nanoparticle. You know that now nanoparticles are used for many kind of application. And so we use again E. coli asparaginase, and the protein was conjugated on the surface of this gold nanoparticle. And also in this case, the amount of protein is really very small with respect to the sample, and so the sensitivity obviously is low. We require very long experiment, but finally we were able to collect good hydrogen-nitrogen cross-polarized HSQC that can be compared with the same experiment collected on crystalline sample and on PEGylated sample of the asparaginase. And then we were able to collect 2D hydrogen carbon plane of 3D HCANH and HCONH. And then we were able to assign 182 amide signal over a total of 224 visible in the spectra. So we were able also to prove again that the protein, when it is bound to the surface of the nanoparticle, preserves its native folding, and we were able also to identify the region of the protein experiencing the largest effect due to the conjugation with the surface of gold nanoparticle. And so this shows how powerful is the solid-state NMR for the characterization of biomaterials and in general of heterogeneous system. I would like to thank you all the colleagues of CERN that collaborated on this project, and thank you to everybody.
Thanks, Marco, for this presentation, very interesting presentation. One comment I can ask you is just how general do you think would be this approach?
So probably the main criticism could be that we have used in many cases the E. coli asparaginase. So what I can say is that we have collected the good results with many different proteins. The main, sometimes we can have a protein with the quality of spectra when you have a small and flexible protein. So I think that this approach is general, cannot be used for all the protein, but I think going ahead with the research and with the experiment, I think that this approach will gain in popularity among the community of the people that are interested in the biomaterials and in heterogeneous system that cannot be investigated nor with solution NMR, and also with cryo-EM. Sometimes it's not easy to analyze, for example, protein embedded in biomaterials. I think that is an important, solid-state NMR can provide an important contribution to structural biology and also to the science of materials.
Questions are coming in. How would you detect the biodegradation of the surface proteins?
Biodegradation, to be honest, for the moment, we didn't observe for the protein that we have analyzed surface degradation. What we were able to, for example, the most interesting results is that in many cases you can say that the protein preserves its folding, and this is not a trivial results, because when you have your protein in within together with other materials, it's not easy to establish that the protein is still folded. But obviously, I think that degradation is a process that can occur in this system. For the moment, we have not investigated this aspect, and we have not observed. What we have observed sometimes is that you obtain terrible spectra. If, for example, the hydration is not enough, or the sample conditions are not good.
A question, can you comment on the preference of using the proton-detected experiments for the assignment?
I think that to assign, you have seen that we have assigned this very big protein, but the assignment was possible integrating, as I told you, for example, the data collected on the protein using the solution NMR experiment for the more mobile part of the protein using a deuterated sample. We have used proton-based experiment for the crystalline sample and also for the PEGylated sample. So are really very important for the assignment of the resonances. Yes.
Okay. In case of proteins bound to nanoparticle, what is generally the measurement time compared to protein sample alone?
I think much more, because the amount is a small fraction. The amount of the protein is a small fraction with respect to the total sample. So, for example, the HSQC you have observed in the presentation were collected in 1 week.
One week, versus when you have the simple isolated protein?
No, in the isolated protein, I think a few hours or less.
Yeah, so from a few hours to a week, so that is...
Yeah, but the amount of protein is really 0.2 milligram.
How unique is the instrumentation necessary to do these experiments?
I can say that to collect this data, we have used 800 and 850 megahertz instruments. And the more important are the probe head. So you need the, we have collected this experiment using a 3.2 millimeter rotor using a ion-free probe, and we have used a 1.3 rotor. So I think that it's not really important if, in this case, obviously high field. But in principle, I think you can get the same results also at 700 megahertz. We'll see what will happen. I'm curious to see what will happen with the 1.2 gigahertz. Also, I didn't show here, but we have a very good preliminary result with the 0.7 millimeter rotor at the 100 megahertz.
A question, just a curiosity on PEGylation. Is it possible to tune the length of the PEGylation?
Yeah, because for example, are available commercially available all size of PEG, because it's of big interest for biotherapeutics. So you can buy. We have used PEG of 1 kD up to 5 kD, 25 kD. There is everything in the market. However, increasing the size, more difficult is the reaction. In our experience, probably we are not so skilled in clever in the conjugation, but we have obtained good results with 1 kD, very good results with the 5 kD. With the large PEG, probably you need a large amount of reagent and so on.
Okay, thanks, Marco. Now I would like to thank all the speakers and all the participants. And I hope you enjoy these few flashes on the activities that are performed at the Italian Center of Instruct-ERIC. And you have seen the instrumentation which is available. And so then I hope you can exploit and get inspired of possible projects that you can do exploiting this type of approaches. And there are many other things. Pulse DPR is another type of applications we can think of, for relaxometer and so on. So then maybe in the future we will present you other types of potential applications, activities that we are doing here in Florence. I give the floor now to Susan from Instruct Hub.
Thank you, Lucia. I'd just like to add to the comments to say that it's been a fantastic webinar today, and it just shows the value and the power of these very advanced technologies that are available at Instruct centers. And I think the Instruct Centre in Florence is one of the showcases of our complement in Instruct. And thank you very much for the great talks today. So the next webinar that we have will come from the Instruct Centre in the Netherlands. And we have 3 fantastic speakers lined up for that. And that will be on the 13th of October. And registration will be open soon. You can also follow some of the discussion and the comments and the activities that arise from these webinars on our Instruct Hub Twitter account. So have a look there, and that will probably give you a little bit of a flavor of some of the discussions that arise from these. And finally, I'd just like to point out that all of these technologies that have been showcased in the webinars, or many of them in any case, are available for access. And if you have an experiment that you would like to use some of the Instruct technologies, then do please register as an Instruct user, and you can access our technologies through this page. Just request access, and you can look at the catalog of technologies that we have in the facilities tab. And at any stage, if you have a question, you can contact us and ask us for advice or information. So do have a look at the website, and we look forward to accommodating you soon. Thank you very much for joining this webinar, and I hope we see you for the next one in October. Thank you.
Thank you, Susan, and thank you to everyone who participated and those who spoke. And I think we can close this now, and we'll meet again on the 13th of October.